In this video, I'm going to talk about ketone body production. Maybe you've heard of the keto diet, or you have heard about checking for ketosis to make sure you're keeping your carbohydrate consumption really low. I'm not going to talk about the validity or utility of any of those things because I'm the wrong kind of doctor for that. My PhD's in biophysics, not in human nutrition. But what I will talk about here is the cool biochemistry that underlies how we actually start making ketone bodies and why that's a really important physiological response in a low carbohydrate condition. So let's dig into that. And I guess the big question here is, what alternate fuel strategies do we use as humans under starvation conditions? So quick reminder that we use different types of fuel that are interchangeable with each other. In some sense, we've got glucose, which can be stored as glycogen, and glycogen, which can be stored as glucose. There's also some sort of sub pieces of glucose, right? We can talk about pyruvate and lactate, which are derivatives of glucose and can be converted back into glucose under certain circumstances. The alternate set of fuel sources we have are built around fatty acids and acetyl-CoA. So we've got fatty acids, we've got their three fatty acids glued together to form triglycerides, and then I'm going to have to hide here. We've got the star of today's show, which I've just shown on a bunch of slides in the past, but I haven't talked about yet, and that's ketone bodies, which are derived from fatty acids, and it turns out are really important under certain types of physiological stress. And what we're going to be thinking about, wrong button. There we go. What we're going to be thinking about today is what happens under starvation conditions. And we as organisms have had to, our ancestors and even some people today, unfortunately, have to deal with situations of food insecurity where there's prolonged periods of time where you can't have access to calories. And we had to adapt. We had to build physiological systems to deal with that. And what we're going to be thinking about is, where do we get the energy that we need to survive given the constraints of our physiology? So first, if you watched my video on fatty acid breakdown, we came across this interesting observation that fat is a very high energy dense material relative to, say, glucose. So if you just look at the normal fat stores we actually have a lot of energy that's available in there. If you don't eat for a long time, you can live for a very long time off of the fat that you have. The other primary store of energy is in our muscles, right? You think about all of the proteins that we have that are built up as muscles. Those proteins are built out of amino acids, and I haven't done videos on those yet, but it's coming soon. Amino acids can actually be converted into sugar and can be used as fat as well. Used as fuel as well. So those are our two sources of energy. And when we're under metabolic stress, where we don't have enough calories, the question is, how do we access these stores so that we can survive? This is made more complicated because different tissues and organs have different fuel requirements. And so this is a table that I'm going to fill in here of different fuel types. So glucose, fatty acids, and ketone bodies. And then we've got different types of tissues, basically. We've got liver, skeletal muscles, the brain, and red blood cells. And it turns out that they have different sorts of pickiness. So the skeletal muscles just doesn't care. It'll use whatever fuel you can throw at it. Whereas red blood cells care a lot. They can actually only live on glucose. And the reason for this is actually red blood cells don't have mitochondria. So they can't do any of the oxidative phosphorylation-y sorts of things. All they can do is glycolysis and lactic acid fermentation. So they absolutely have to have glucose as their fuel source. They can't use anything else. There's a few other tissue types like that. For example, your lens cells have almost no mitochondria. They do this really interesting differentiation process where they basically demetabolize themselves. And there's very low oxygen availability in your lens. So again, they need glucose. They can't use other fuels. But then you have things like your brain. In your brain, the reason it can't use fatty acids, not because it doesn't have the machinery to eat fatty acids necessarily, but because you have the blood-brain barrier, right? This hydrophobic barrier that separates your brain from your bloodstream. And so fatty acid just can't get across that barrier. So instead, your brain prefers glucose, that's its preferred fuel, but it can also use these ketone body things we're going to talk about today. And then lastly, the liver is interesting because the liver can happily consume glucose and fatty acids, but it actually can't consume ketone bodies. And the reason for that is it makes ketone bodies. And to be a net exporter of ketone bodies, the last thing you want to do is break it down. So what's our problem? So we're trying to ask the question, well, what happens when you have a low you're under starvation conditions, you're not consuming calories. You've got this store of calories in fat and the store of calorie in muscles, and then you've got all these tissues that have different requirements. So how can we survive even with these picky eating tissues? And it turns out that there's two processes that we can that we do to solve the real problem of how real problem here, which I guess I should say the real problem is how do you get enough glucose and ketone bodies, right? Because fatty acids, they're the last thing that's going to run out, right? If you've got fat stores, you can continually make fatty acids. So tissues that can live on fatty acids are going to be fine. What we've got to figure out how to do is how do we feed our brains and our red blood cells under low energy conditions? And it turns out the answer to this is a combination of gluconeogenesis and ketone bodies. Okay, so let's think about the starvation conditions in our very schematized cell here. We have our muscle cells, which are happily burning fatty acids. We have our adipocytes, our fat cells, which are liberating their fatty acids. I'm going to make myself go away actually. They're liberating their fatty acids, so they're providing fuel. Our blood glucose is low because usually when I draw this picture, I've got glycogen in there. So our glycogen is gone. So we need to be somehow replacing this glucose with something. And it turns out the way we're going to be doing it, the first line of defense, or the first thing we're going to be doing is running this process of gluconeogenesis. And so what's happening in gluconeogenesis, if you haven't seen my video on that or you don't recall, you can go look it up. But basically the idea is you take molecules like pyruvate down here. This is a 3-carbon sugar, the end point of glycolysis. And we are going to take pyruvate and we're going to run it on a wending path backwards from pyruvate to oxaloacetate to phospholinopyruvate and then back through. This is glycolysis, just kind of running backwards up here and then do a couple of alternate steps. But ultimately, we can make glucose out of our pyruvate. And this is great, right? This means that we can keep our blood glucose up. And if we keep our blood glucose up, then we can say feed our red blood cells and our brains. What goes in as fuel? Well, the fuels for this are amino acids and lactate. So these can come in as pyruvate or oxaloacetate, right? So if you're breaking down your muscle tissue, it's going to be coming in as one of these molecules, and then that can be used to make glucose. So, I mean, it's never good to be breaking down muscle tissues for fuel, but if it's the difference between staying alive or not, this is a really great strategy to keep our blood glucose up. Okay, so what we're saying there is we are dumping out glucose by gluconeogenesis. What about fat stores, right? Could we use our fat stores to help keep our blood sugar up? And that would be awesome, right? Because we got a lot of energy in fats. Why not just use that to make glucose? But the problem is we literally can't do that. And we do the next best thing as a response, and that is make these things called ketone bodies. So how does that work? So fatty acids, remember, are broken down by this process of beta oxidation. And so here I've got our favorite fatty acid. This is, came out of our fat cell. We're going to break this down into a bunch of acetyl-CoAs. So if you take a fat and you break it down, the output of it is 8 acetyl-CoAs. And the idea would be maybe we could take an acetyl-CoA and feed it into, say, the citric acid cycle. And if we do that, maybe we can figure out a way to make oxaloacetate, right? I don't know if you have your I can't talk and write the same, but oxaloacetate. I don't know if you have your citric acid cycle memorized, but oxaloacetate's in the citric acid cycle. So, that suggests maybe you could take an acetyl-CoA, throw it into the acetic acid cycle, make oxaloacetate. That could go on and make glucose, right? That would be great. But unfortunately, this cannot be done. And the problem is the citric acid cycle actually can't make acetyl-CoA into oxaloacetate. So let's look at that. So I'm going to show this zoomed up for a second. This is our first step in the citric acid cycle. This has an oxaloacetate and a citrate. And the acetyl-CoA comes in, drops off its acetyl group that gets stuck onto the oxaloacetate to make citrate. Okay, we made citrate. How do we make oxaloacetate? Well, we just go backwards. If we run it backwards, oh shoot, we regenerated our acetyl-CoA, right? There's no way to, at this first step, This step is driven forward by taking the acetyl group off of the CoA and sticking it onto the oxaloacetate. And if we go backwards, we're just pulling the carbon off or the acetate off. So we're not really we can't put the acetyl group in and still have oxaloacetate come out. Okay, well, excuse me. Maybe what that suggests is we should go all the way around. Instead of doing one going this direction, we should go in this direction all the way around, get back to oxaloacetate. That makes sense, right? But there's a problem. That problem is both of the carbons from acetyl-CoA are lost as carbon dioxide. So if you look at this, right, we have acetyl-CoA coming in, 2 carbons, 1 carbon, 2 carbons. Those get kicked off as acetyl-CoA, as carbon dioxide. And if you recall the net reaction for the citric acid cycle, it's literally acetyl-CoA plus a bunch of things like NAD plus goes to carbon dioxide plus CoA plus a bunch of other like things like NADH, right? It's gone. The carbons are lost. And what that means is if we say dump acetyl-CoA in, and then we pull oxaloacetate out as a way to go, I don't know, run the run gluconeogenesis, we're actually going to cause the citric acid cycle to halt. Because this molecule, as it's getting pulled out, we're depleting the citric acid cycle. Every time we run it, and then we pull it out. Now we can't restart the cycle, right? We've taken out the starting point for the cycle. It's now no longer a cycle. It's become a directional flow, a pathway that's taking oxaloacetate out. Okay, so we can't use acetyl-CoA to make oxaloacetate. And if we pull oxaloacetate out of the cycle, it stops. And it turns out that actually happens. And maybe you recall, maybe not, but oxaloacetate actually has the lowest concentration of any of the metabolites in the citric acid cycle. So this is taken from bacteria cranking along at full aerobic respiration, so it might be a little different in the human mitochondrion, but still, its concentration is 0.004 millimolar. Contrast that with malate, which is 1.1 millimolar. It's like 1,000 times lower concentration than many other metabolites. What that means is it's actually quite sensitive to depletion. And when a liver cell is, say, running gluconeogenesis, it's actually going to deplete mitochondrial oxaloacetate even lower than what we showed here. So I'm going to use the transit diagram as the way that I've been wanting to use it since I started making these videos. So right here, this is the process of gluconeogenesis. Let's zoom in. Ooh. We started with pyruvate. We go to oxaloacetate, go to phosphoenolpyruvate, and then go all the way up to glucose. Cool. How do we connect into that from the citric acid cycle? Well, look, there is an oxaloacetate there, right? This is inside of, I should orient us to the citric acid cycle. So we have acetyl-CoA coming in to oxaloacetate to form citrate, and then this is the cycle going around. Well, it turns out oxaloacetate does not have a transporter in the mitochondrial membrane, inner membrane, but citrate does. So oxaloacetate goes to citrate, takes in our acetyl-CoA. That gets exported back to citrate, and then into acetyl-CoA. Into oxaloacetate, and then into acetyl-CoA in the cytosol. But what we're basically doing is if we're running the citric acids, if we're running gluconeogenesis hard enough, if we're dragging enough mass this direction, if our flow is in this direction all the way, excuse me, what that is going to lead to is a depletion of citric acid cycle intermediates, and it's going to lead to a specific depletion of this oxaloacetate because it had the lowest concentration to begin with. So when a liver cell is running gluconeogenesis, it depletes the mitochondrial oxaloacetate even further. That means that the citric acid cycle slows down, which has 2 consequences. Pardon me. The first consequence is you get lower energy production. That's not a good thing, right? We have lower energy production. Overall, that's generally not helpful. But the second, maybe more catastrophic thing is we actually run out of coenzyme A. Think about it. We're breaking down all these fatty acids, right? We have no sugar around, so we're just breaking down fatty acids. That's our energy. We're making a ton of acetyl-CoA. All this acetyl-CoA is here. Each one of these has a coenzyme A attached. And we just ran into a situation where we can no longer run the citric acid cycle, right? Our CoAs are coming up to the citric acid cycle, and it's stopped. So these CoA concentration, or the acetyl-CoA, sorry, concentration starts to go up. How do we fix that problem? We fix that problem with ketone bodies. Enter ketone bodies. And ketone bodies are synthesized directly from acetyl-CoA. And I'm showing the three most common here. This is the three most common. But hopefully you can appreciate these all look a lot like acetyl-CoAs. Or sorry, acetyl groups. They're basically acetyl-CoAs glued together in interesting and different ways. What this does is it has several consequences. First, it frees up coenzyme A. So we're doing, as we're running gluconeogenesis, we're depleting the citric acid cycle. We can free up acetyl-CoA or CoA molecules by a different route, that is by making ketone bodies. The second thing it does is these ketone bodies can be exported into the bloodstream and used as fuel. And so we can add that to our picture here. So we have our liver cell, which is running gluconeogenesis. It's breaking down things like muscles to generate proteins to make glucose. That now triggers a response where liver cells import triglycerides, convert them into ketone bodies, and export those ketone bodies. And then those ketone bodies can be used by other tissues as fuel. So it's basically a different, it's a different level of fuel, right? We normally have fatty acids and glucose. Now we've added this third thing, this ketone bodies, into the mix. Inside of a consumer cell, if it's not obvious, the cells will take that ketone body, convert it back into acetyl-CoA, and then they can use that in their own citric acid cycle. So a muscle cell isn't running gluconeogenesis. It's not doing this depletion thing. So it can happily run its citric acid cycle over here, and it can then thus use the ketone body. Overall, what this means is that cells that need glucose can use the precious supply. So red blood cells, they can't use ketone bodies. They don't have mitochondria, right? But they can use the glucose that's being made by gluconeogenesis. The brain, on the other hand, it can use ketone bodies because they're small and can diffuse across the blood-brain barrier. So when glucose concentrations go down, ketone bodies can start to fill in that gap. So what that means is under low, extremely low carbohydrate conditions, you basically have your liver can just operate on fatty acids. Your muscle cell can operate on fatty acids and ketone bodies. Your brain can operate on ketone bodies, and they still need some glucose. And then your red blood cells can focus on using glucose. So this ketone body supply is really important to allowing correct distribution of resources in these resource scarce conditions. Okay, so let's summarize what we've talked about here really quickly. What we said is that we can keep our blood glucose high for certain cell types, or we have to keep our blood glucose concentration high for certain cell types, even if we're not consuming carbohydrates. We have no carbohydrates coming in. What do? Well, we have to do something. We can't directly convert fatty acids into glucose because the problem in the citric acid cycle, right? Acetyl-CoA is converted to carbon dioxide. It cannot be used to make something like oxaloacetate. So what that means is our liver cells continually running gluconeogenesis to make blood glucose, but that's depleting oxaloacetate from the citric acid cycle and causes acetyl-CoA to build up. What this triggers then is this cool response where we now make ketone bodies, which frees up the CoA and then provides an alternate fuel that we can use in these conditions of low carbohydrate. Okay, so that's what I have on ketone body production. Hopefully it was interesting and helpful. I look forward to seeing you later, but for now, I will talk to you later.